Welcome. You're watching Certara Talks. I'm Dr. Suzanne Minton, and I'm a director of content strategy at Certara. Today, I'm talking to my colleague, Dr. Krista Greenwood. Krista, welcome to Certara Talks.
My name is Krista Greenwood. I'm a director of toxicology here at Certara, board certified toxicologist.
Awesome. Well, great to talk to you. I hear you just came back from the American College of Toxicology meeting. Can you let us know how it was?
So it was in Orlando, Florida this year. It's the annual meeting. It is a really great organization that is a little small. There's maybe 1,000 attendees, but very focused on drug development toxicology, which is a really great fit for myself and my colleagues in the toxicology group.
Fantastic. Well, I was hoping we could chat about some of the themes at this year's meeting. Project Optimus, the FDA's initiative for dose optimization of oncology products, has been a big theme in the industry as a whole. What did folks at ACT have to say about Project Optimus?
Yeah, so there was actually a session on Project Optimus, which I attended, and it was very well attended. And there was a lot of talk. It was more on the clinical pharmacology side and, maybe that clinical side. But at least from a non-clinical perspective, where I'm coming from in toxicology, it helped to understand kind of the end game and the point of why we might want to try to, optimize doses in the clinic. And then just give that perspective to us as non-clinical toxicologists that are, all of our research is occurring far before things get into the clinic, but it's still nice to have that end in mind.
Absolutely. I'm not a toxicologist, but I certainly, from what little I know in the field, I know that the 3 R's have been a long-standing theme. This is the idea of reduce, replace, and refine the use of animal testing in drug development. What were people at ACT saying about the 3 R's this year?
So the 3 R's were a hot topic ever since COVID. There's been a short supply of both non-human primates and now even dogs, some of the non-clinical models that we use. And so it is at the front of everyone's mind to really make sure we're using animals in a responsible way because they are such a limited resource at this point. And, there was a lot of discussion about the best way to design studies, uh, opportunities to use historical control data more than what we are using it currently. So I'm hopeful that in the near future, the regulatory agencies will really support us as toxicologists in making the change and really reducing those number of animals that we use.
Yeah, that would be a great thing to help reduce the amount of animals required for these programs.
Mm-hmm.
Lastly, I heard that there were some updates to the ICH E14 S7B best practices guidance. Can you tell our listeners what that guidance is and what they should know about it?
Right. So, just in general, it's a mouthful, but it was an update to the best practices for the safety pharmacology, specifically the cardiovascular aspect of the safety pharmacology guidance from the FDA. The hope is that we can reduce the need for human thorough QT studies, and so some of new best practices came out. And there was a lot of discussion about how to implement these in the non-clinical studies that we perform, things like the HERG in vitro study and the in vivo cardiovascular study.
Fantastic. Well, Krista, it was a real pleasure talking to you today. If our listeners are interested in learning about more about what Krista and her team do, they can always visit us on the Certara website. You've been watching Certara Talks. I'm Suzanne Minton. I'll see you next time.